Hello, welcome, and thank you for attending today's webinar entitled The Power and Simplicity of Oocyte Recording with the TEV-700 Oocyte Workstation. My name is Edmund Buck, and I'm a product manager here at Warner Instruments. In today's presentation, our speaker, Dr. Cecilia Canessa of Yale University, will demonstrate how easy it is to acquire data from oocytes using the TEV-700. In addition, she'll review several interesting recording techniques she's used over the years, and will share some interesting findings that came from using those techniques. And finally, the presentation will be followed by a question and answer period. Before we begin, I'd like to address a couple of housekeeping items for today's webinar. First, this webinar is being recorded, and after the event, everyone will receive a link to the recording via email. So don't worry if you miss anything along the way, since you can review the full presentation again at your leisure. Second, if you'd like to ask a question during the event, then feel free to type your question into the chat box located on the bottom right-hand side of your screen. At the end of Dr. Canessa's presentation, I'll read any submitted questions aloud for her to address. At that time, I'll also be available to answer any questions you may wish to ask of me. With that being said, I'd like to introduce today's speaker. Dr. Cecilia Canessa is a professor of cellular and molecular physiology in the Department of Cellular and Molecular Physiology and professor of medicine in the Department of Internal Medicine at Yale University in New Haven, Connecticut. She received her MD in Lima, Peru, and has been at Yale since 1994. Her primary research interests have focused on understanding the structure, function, and regulation of ion channels from the ENaC degenerin family in the kidney and peripheral and central nervous systems. Much of her work has been groundbreaking, and she'll be sharing some of those techniques with us today. Before handing the microphone over to Dr. Canessa, let's begin by watching a short video where she demonstrates the ease of working with the TEV-700.
So I'm going to introduce you in this short film about the two-electrode voltage clamp. This is a very useful technique when you want to characterize proteins that are electrogenic. There are only two conditions that need to be met. The protein of interest has to be expressed at the plasma membrane, and the other condition has to be electrogenic. So it's good for study ion channels, transporters, ion pumps. The equipment is designed to be used with the oocyte expression system because the cells have the advantage of being very large. They can be micro-injected 1 or 2 days prior to the experiments. And then the technique is extremely easy to master. And it basically consists on placing your cell that already expresses the channel or interest in this small chamber that is constantly perfused by the solution of your choice through this perfusion system. Now, you can have also a vacuum or like you can see here, or you can connect it to the vacuum of your laboratory. Once the cell's in place, in order to do the experiment, you have to impel with 2 electrodes that are handled by these micromanipulators. The signal recorded then goes to this amplifier, where you can control the voltage and also do some manipulations, like for instance, you can run a ramp, a voltage ramp, and then you have an acquisition data that feeds to your computer where you record and store and even do some manipulations with the data. In the next part of the film, we're going to show you exactly how the technique is done and how actually easy to train an individual that has never done it in a few days will be ready to produce reliable data. So now I'm going to show you how to conduct an experiment. So the first thing we take is one oocyte and put it in the chamber. All right. So now we introduce the two electrodes, voltage and current electrodes, into the bath solution. And we look at the voltage difference of the two electrodes. It should be 0. And we adjust that with the offset electrodes until they reach the value of 0. Then we want to test the resistance of the electrodes. They should be less than 1 mega ohm. And we do that by pressing on the electrode resistance test. If they are of the correct value, then we advance the electrodes close to the surface of the oocyte until they form a dimple. At this point, we advance slightly the two electrodes until we penetrate the plasma membrane. Now we should read a voltage that it's the difference between the intracellular voltage and the bath. Now the oocyte has been impelled and is voltage clamped at minus 60 millivolts. And we do not observe a current because the channel is not activated. In order to activate it, I'm going to change solutions to a lower pH. And that's the current that we obtain with pH 6.8. We'll return to pH 7.4. Okay, so once the experiments with the oocyte is completed, we unclamp the oocyte, remove the electrodes, and we can place a fresh oocyte. As you can see, the experiment can be very short, few minutes, and in a whole morning you can complete an analysis of 10 to 20 oocytes. Okay, in the next segment of the webinar, I will present examples from our own laboratory to illustrate uses of the two electrode voltage clamp. First, a few general principles. As I mentioned before, this technique enables you to examine any electrogenic plasma membrane protein. By electrogenic, I mean that the protein of interest has to transport a net charge across the membrane. The identity of the protein doesn't matter. It can be an ion channel, a transporter, or an ion pump. I will show examples of each of these classes of proteins during the presentation. This is a list of studies we have conducted in my laboratory using the Xenopus oocyte and the two electrode voltage clamp for recordings. First is functional cloning. We use this approach to clone ENaC, that is an epithelial sodium channel, at a time when we didn't know the molecular identity of the channel. Although to date, the whole genome has been sequenced and annotated, there still remain instances where you need to identify the protein underlying a conductance in a cell of interest. The success of this approach relies on selecting a distinctive signature of the conductance that will be used through the screening process. It can be a blocker, an activator, or something else. I will expand on this later. A more frequent situation we face to date is the case of functional differences when a channel is examined in a tissue of origin their difference compared to the expression in a heterologous cell. One explanation is that the channel is missing a modulator or an auxiliary subunit, which can be found by the cloning strategy of functional complementation in oocytes. The most common use or application of this technique is functional characterization of a channel or transporter. One can examine kinetics, determine ion selectivity, measure affinity of agonists or inhibitors. It is also useful for screening small molecule libraries or complex venoms, looking for activators or inhibitors of an ion channel. You can also conduct a structural functional analysis of your protein by examining the functional consequences of amino acid substitutions. Measurements of rates of cysteine modification by thiol-specific reagents give information on conformational changes of the protein. Similarly, voltage clamp fluorometry measured changes in fluorescence intensity induced by a perturbation. From this type of data, we can infer about conformational changes of the protein. This is just a short list of applications. New methods continue to be developed, and the limit is your imagination and ingenuity to answer specific questions. The technique has some restrictions of limitations. The protein to be examined has to be expressed at the plasma membrane. If your protein of interest expresses in an intracellular organelle, for instance, the ER, lysosome, or mitochondrium, you might need to isolate that membrane and use, instead of the 2-electrode voltage clamp, the preferred technique will be the patch clamp. Otherwise, you can purify the protein and reconstitute it into lipid bilayers. Another drawback of the 2-electrode voltage clamp is that you don't have access to the intracellular side of the protein. There are some rapid events in the range of milliseconds induced by changes in composition of the bathing solution that also can be missed. Features of the unitary channel currents are not detected. There is also the need of an animal facility equipped to keep frogs, and you need to apply for an IACUC permit, which can be really a lengthy process. However, recently, these inconveniences have been eliminated by companies that deliver Sinopus oocytes to your laboratory. Now, a brief overview of the procedure. First, you isolate the oocytes from the frog and inject mRNA extracted from tissues or cRNA prepared from plasmids containing the coding region of the protein of interest. The RNA is injected in the cytosol of the oocytes in a volume of less than 50 nanoliters. This is by far the most common procedure. Some researchers prefer to use DNA, but then the nucleus is injected, which is technically more difficult. After the injections, the oocytes are kept at 16 degrees for variable incubation times, usually 1 to 5 days. The optimal incubation time to reach maximal expression levels varies and need to be determined for each protein. This slide lists the steps for functional cloning using ENaC as the example. First, it involved the construction of a cDNA library from mRNA of a tissue that expresses the channel of interest. In our case, we use epithelium from the distal column from rats kept in a low-salt diet in order to maximally increase the abundance of ENaC mRNA. The library is divided in pools of 10,000 to 8,000 clones, and cRNA is prepared from each pool. Then you inject 10 to 20 oocytes from each of the pools of cRNA and screen individually the oocytes with a 2-electrode voltage clamp using a signature of your protein. In our case, we use the ability of amiloride to block the inward current of ENaC. If you get a positive group of oocytes, then you select that pool of the library and divide it in smaller fragments and repeat the steps several times until you get to a single clone. In this slide, in the left upper panel, you see representative currents of ENaC expressing a single subunit of the channel, the alpha subunit. The magnitude of the current is very small, only 10 to 15 nanoamps, and it's a constitutive conductance that would be missed in the background of the oocytes. To isolate it from the oocyte endogenous currents, we use the specific blocker amiloride, 5 micrograms, indicated by the black bar over the current trace. Even this small but specific signal allows the isolation and cloning of the first pore-forming subunit of the channel. The example clearly illustrates the power of the technique. The other two subunits, beta and gamma, were cloned by complementing alpha ENaC with the original library. As shown in the right upper panel, addition of beta and gamma subunits to alpha ENaC increased the magnitude of the current by more than tenfold. In the left lower panel, we see the block of ENaC currents by amiloride. And with this data, we can then calculate the apparent affinity for amiloride block. The IC50 is 110 nanomoles with an N coefficient of 1. We also use the 2-electrode voltage clamp to demonstrate how some ENaC subunits found in the carboxy termini of beta and gamma subunits in patients with Liddle syndrome produce high blood pressure. ENaC is expressed in the apical side of cells in the kidney where sodium is absorbed. Too many channels lead to too much sodium and water intake into the blood, resulting in hypertension. Using the patch clamp technique, lower panel, we found an increase in number of channels at the cell surface expressing little mutations, but there was no change in the conductance or in the open probability, suggesting that the number of channels was at fault. Here we see in the left upper panel that the mutations or truncations of the carboxy terminus of beta and gamma subunit increase ENaC currents in oocytes. If we look now at the left lower panel, and if we add Brefeldin to the incubation medium. Brefeldin is a drug that blocks delivery of proteins to the plasma membrane. The wild-type INAC current progressively decreases, consistent with the notion that channels are being removed from the plasma membrane by endocytosis. That's the red line. In contrast, channels with little mutations, look at the blue line, seem to be stable despite Brefeldin, suggesting that they are not endocytosed as rapidly as wild-type channels. To examine further whether endocytosis plays a role, we co-injected a negative dominant variant of dynamin, shown as dynamin K44. This molecule uses ATP to sever endocytic invaginations from the plasma membrane, as the cartoon in the right upper panel shows. Co-injection of wild-type INAC with dynamin 444 increases INAC currents, but it doesn't do in little syndrome mutants, as shown in the right lower panel, which is consistent with the notion that the C termina contain endocytic signals that if removed, lead to accumulation of channels at the cell surface and an increase in sodium uptake. This example illustrates that a whole biological process can be reconstituted in the oocytes by injecting 4 to 5 different cRNAs and following the activity of the channel with a 2-electrode voltage clamp. Now I'm going to turn to ASIC, that is an acid-sensitive ion channel. ASIC has the same structure as CINAC, therefore belongs to the INAC degenerate family of ion channels. However, ASICs are very different in function, mode of activation, and are expressed in neurons of the central and peripheral nervous system, but not in epithelial tissues. These are currents that show two main properties of ASIC channels: activation by external protons in the upper panel, and steady-state desensitization by protons in the lower panel. In this experiment, the membrane potential of the oocytes was held at -60 millivolts, and the external solution containing 120 millimolar sodium was rapidly changed from 7.4 to an activating solution of low pH, as indicated by the bars above the current traces. In this example, protons evoke large inward currents that desensitize in the continuous presence of external protons. The peak current evoked by pH 6 is around 45 microamps. Taking the values of the peak current at the indicated proton concentrations, we can calculate the apparent proton affinity for activation as shown by the green line in the right panel. The EC50 is pH 6.6, equivalent to 270 nanomole of protons. On the other hand, if we keep the activating pH fixed at 6.0 and sequentially decrease the pH of the recovering solution, look at the lower trace, one observes progressive decrease of the peak currents. This phenomenon is due to channel desensitization. The EC50 for proton-mediated steady-state desensitization was calculated to be 7.1 or 79 nanomoles of protons, as shown by the blue line. This example illustrates how one can examine important physiological properties of an electrogenic protein with a 2-electrode voltage clamp. Now, suppose that you want to explore conformational changes of a protein of interest induced by a perturbation, either adding a ligand, a blocker, or a change in membrane voltage. Carlin developed in 1992 a method that proved to be very efficient and easy to use for this purpose. Let's provide a brief explanation of the method for the novice in the audience. It consists in introducing a cysteine residue in the region of the protein that will be tested for conformational changes. Other cysteines in the protein ought to be removed to avoid confounding results. If the protein tolerates such a mutagenesis, you can use the method. The second step is to measure the apparent reaction rate of the methane thiosulfonate reagent by monitoring current changes while the reagent is applied in the resting state and again during the stimulus. The underlying principle is that if the cysteine is hidden from water in the resting state prior to the perturbation, the rate of the reaction will be slow. If the perturbation exposes the cysteine to water, then the reaction will be much faster, or vice versa. There are several assumptions and caveats important to the interpretation of these experiments. To be aware of them, I recommend reading the original papers. These reagents come in various sizes and charges, positive, negative, or neutral. Therefore, if the initial chosen reagent doesn't work, you can try a smaller one to fit into the area where the cysteine has been introduced. If the modification doesn't change the current, little can be concluded. There are many reasons for such a negative result. Only when the current is modified by the reagent, one can build a hypothesis. This technique can be used to examine the extracellular domain and transmembrane segments of a protein using the 2-electrode voltage clamp. Here I present an example. This is ASIC1 modified by MTSCA in position asparagine 415, which is located in the extracellular domain. N415 was replaced by a cysteine. No other free cysteines are present in the extracellular domain of ASIC. Then it was exposed to 10 micromolar of MTSCA, and we can see that this slows the rates of activation and desensitization, upper traces. There is also changes in the apparent affinities for activation and steady-state desensitization that are shifted to the right, shown in the left lower panel. However, the rates of MTSCA modification are not significantly different, whether it is applied in the closed or desensitized states, as shown in the right lower panel. From these results, these results suggest that the position N415 participates in conformational changes that open and close the channel, and it is accessible to water in both the open and the closed states. This interpretation was indeed confirmed a few years later when the atomic structure of ASIC was solved in both the closed and the open states. The figure shows superimposed structures of the desensitized conformation in gray and the open conformation in orange. For simplicity, only two subunits appear in the figure. Residue N415, shown by the arrows, is located in the short linker that joins the beta strands 11 and 12. Notice that the residue N415 flips in the transition from open to closed. It faces outwards in the desensitized state but faces towards the interior of the cavity in the open conformation. A few methane thiosulfonate reagents are membrane permeable and can reach the intracellular side of the protein, but one loses control of the concentration. In this case, the patch clamp in the inside-out configuration is the alternative choice. Here is an example where we apply the technique to the second transmembrane domain that forms the ion pathway of ASIC. We complemented results of the 2-electrode voltage clamp with patches in the outside-out and inside-out configurations to have access to both sides of TM2. The figures show the rates of modification with MTSEA of each residue replaced by a cysteine in the open, red and orange symbols, and in the closed, blue and green symbols. From the data in the previous slide, we can reconstruct the architecture of the pore in the closed and the open states. These results were again in agreement with the crystal structure published recently. We can see there how the transmembrane domains adopt different conformations. Now, let's change gears and present some data of a transporter. In this case, is the sodium iodide symporter, also known as NIS, that was originally cloned using a functional assay in xenopus oocytes by Dr. Nancy Carrasco. This molecule is found in the basolateral side of the epithelial cells that form the thyroid gland, where it transports one iodine ion and two sodium ions from blood into the gland. Thus, it generates an inward current. Other anions besides iodide are also transported. Periodate, thiocyanate, nitrate, and periodate, but not perchlorate. This slide shows in the left upper panel currents of NIS. 300 micromolar iodide evoke an inward current, but not 100 micromolar of perchlorate. Mutations in NIS by Dr. Carrasco's group make possible to transport perchlorate, current trace on the right. Notice that the magnitude of the current here is less than 1 microamp, much smaller than in the previous examples, owing to the slower ion transport rate of transporters compared to ion channels. The 2-electrode voltage clamp can be used together with fluorescent measurements to detect conformational changes of a protein upon a perturbation using a technique now known as voltage clamp fluorometry, which was pioneered by Dr. Isakov. Here, a thiol reactive fluorophore labels a unique cysteine placed at a strategic position in the protein. The fluorophore is sensitive to the surrounding environment. Therefore, it can report conformational changes of the region of the protein containing the cysteine. The direction of the fluorescent change, up or down, is irrelevant. What is important is to observe a change in the intensity of the fluorescence. Routinely, endogenous cysteines on the surface of the oocyte are first blocked to reduce background, and cells are incubated for 1 to 3 days waiting for expression of the injected cRNA. The cysteine in the newly synthesized protein is then labeled as shown in the left upper panel by those red dots. These experiments require additional equipment besides the 2-electrode voltage clamp and is indicated in the right upper panel. Mainly, you need an upright fluorescent microscope equipped with a 40x water immersion objective for fluorescent recording. High-quality fluorescent filter sets, excitation, dichroic, and emission appropriate for the fluorophore used. A triggered shutter to avoid unnecessary bleaching of the fluorophore. The left lower panel shows representative changes in fluorescent intensity evoked by a blocker of the transporter and measured at different voltages. This result indicates that the cysteine in one of the transmembrane domains senses both conformational changes induced by the blocker and by changes in membrane voltage. Usually, such a positive result is followed by placing the cysteine in several neighboring positions to build up a hypothesis about the conformational changes of the region of the protein. At this point, I thank you for your attention, and we will have opportunity to answer some of your questions.
Thank you very much, Dr. Canessa, for an informative presentation. Clearly, there are a large number of techniques that can be brought to bear on oocytes using the 2-electrode voltage clamp. We would like to now turn to answering questions from the group. So during your presentation, there was a little bit of discussion on a commercial source for oocytes, and 2 companies were offered up. And I think the group can see those. My question to you is for those who don't wish to buy oocytes, how hard are they to grow and harvest from the frog?
Actually, it's extremely simple. The only inconvenience is that you have to apply to your university for a IACUC permit. That can be a lengthy process. By that I mean can be a couple of months. Most animal facilities can keep frogs because they are very easy to maintain. And the operation of the frog, that is an ovariectomy, it takes about 10 minutes. So, and at that point is exactly what you get from the companies, a piece of the ovary. Does it answer your question?
I believe, yes. One of the questions that I have is, what is the endogenous conductance in oocytes in the absence of expressed channels?
Besides the large size of oocytes, one advantage is that the endogenous conductances are minimal. The oocytes are very silent. That means that you have a very little background when you express your protein of choice.
Okay. When you have endogenous currents, are there proven techniques for controlling those currents so you don't have overlap with the expressed channels?
Actually, there are. And people that have been working with oocyte for a long time learn to know what is in the membrane of oocytes. And I can tell you there are very little water channels, almost none, so you can change the osmolality of the solution for relatively short periods of time, and maintain a perfect stability of the cell. Ion channels are minimal. There are a few that can be opened with certain maneuvers. For instance, if you remove calcium from the bath, you can open some connections that will generate an inward current. But the calcium concentration to activate such endogenous connections has to be very, very low in the micromolar range. So it's good to know that. You can even block some endogenous conductances if you inject some antisense oligonucleotides that will eliminate those currents in 2 to 3 days. There's also an endogenous sodium-potassium ATPase that generates a current that is in the range of up to 200 nanoamps of current. Because the oocyte has a very high sensitivity to wobbeing in the nanomolar range, if you want to express a sodium-potassium ATPase from rat or any rodent, you can block the endogenous by adding nanomolar concentration of wobbening, and still you can examine the injected cRNA that expresses the hump from rat, for instance. There are many tricks that can be used. I hope it helps.
Okay. One of the questions from the group was, what data acquisition system and data analysis software do you use in your research?
We have used over time many things, but most people use the Axon software, or we have used also HECA, and also we have used AD Instruments lab chart that is very good when you have slow, when you are recording the slow events. So lab chart is really good.
Okay. Another question from the group is, for fast ligand-gated channels, it's common to measure both the peak current and area under the curve. Have you compared these two parameters in your ASIC systems?
Yeah.
With your ASIC channels? Mm-hmm.
Of course, because the channels desensitize, and the rate of desensitization is varies according to the protein. We can measure both, and that's done by the software.
Okay. Another question that's come in is, how do you clamp the voltage under currents of tens of microamperes?
Well, if you get, as I show you one example, we have in certain occasions very large currents. Ideally, the currents should not be so high because it becomes difficult to clamp. So this channel that I show you, that is ASIC as well as INAC, are not voltage dependent. If the currents are too large, we need to clamp to diminish the current. We clamp at -40 or -30, and we always follow the channel of voltage clamp to be sure that it's very stable. If it's not stable, that oocyte that measurement cannot be taken seriously. It's unusual, the fact to have very high currents. Most of the time we deal with small currents, but ASIC expresses very well, especially the human one.
I see. In follow-up to the question about the endogenous currents, one of the group members mentioned that the endogenous conductances are in the order of a nanosiemens compared to hundreds or thousands of nanosiemens with the ENaC channel. Can you speak to that?
Okay, I'm not exactly sure if I got the question right, but
Basically, I would just was hoping that you would verify that the person's comment was that the scales are so much different that the endogenous current is not particularly a problem.
okay. I show the example of ENaC expressing only a single subunit because it really demonstrates or illustrates how much you can do with this technique. When we were cloning the channel, the first subunit generated a very small current. And it's an inward current that transports sodium. And the channel is constitutively active. So obviously 50 nanoamps of current or 20 nanoamps, you cannot see them. They are blended in the endogenous currents of the oocyte. The only way we were able to separate it out was by adding a millurite to the bath. No, at the very small concentrations of a millurite, like 1 micromolar, don't block any endogenous currents of the oocyte. So if you can see the block, that means that you are expressing ENaC. And that was the success of the technique, having a very specific way to identify the current of interest. But can be anything. I mean, if your channel or your protein is sensitive to a toxin, you can use that.
Okay. I see that the group is very interested in purchasing oocytes. So, are there any more comments for the speaker? Any more questions for the speaker before we move on to the next slide?
Okay, there are several companies that offer this service. We have used some of them, and really they are good. They deliver almost a whole ovary in a 50 ml Falcon tube. You place the order, that has to be done at, I think it's several days in advance. They come by FedEx to your lab, and you have to prepare the oocytes because they are enveloped in the membranes of the ovary. So you have to separate them out, treat them with collagenase, and that's it. So they're in pretty good shape, actually.
There was another question that just came in for Dr. Canessa. Have you done relative ion permeability studies?
Yes, yes, and because we don't have access to the interior of the cell, we don't know exactly the composition of sodium, potassium chloride, of any ion. So we just have to change the external solution, and with that we measure with ramps the reversal potential, and then we can get relative permeabilities of ions just by looking at how the reversal potential changes. You just apply the Nernst equation. Of course, in the patch plant is different because you control the composition of the pipettes and the bath. So.
Moving back to the expression of channels in the oocytes, and this is while other people are typing. I understand that, or I recognize that cells, they must be injected in order to express the channels. How accepting are the cells to this injection procedure? And more to the point, what's the usual success rate for injecting and getting expression in the cells?
Okay. Um, in general, you can express anything in the oocyte. There's a certain degree of toxicity, but usually when the channels are constitutively open. Most channels require some activation by a ligand, a change in voltage, or some maneuver. And if you don't do that, the channel or the protein can be a transporter, like I show you in this, is closed because there's no iodine present in the bath. In the case of ASIC, if the pH is about 6, you don't get any current. There is a problem with ENaC because that is a constitutively open channel that conducts sodium. If you leave it that way for days, the cell is going to swell and become in really bad shape. So what we used to do is to add amiloride, a blocker amiloride, to the bath to prevent toxicity. And by that simple maneuver, the oocytes can last 2 to 3 days. No, not too much longer, especially if there's very good expression.
Very good. So are there any more questions from the group for today's speaker? I'll give the people typing an opportunity to type. A question that's come in is how much RNA is injected to express in 1 to 2 days?
Okay, the amount of RNA injected also varies, but in general, 5 nanograms is more than enough. And the time that needs an oocyte to be incubated varies with the protein too. For us, both ENaC and ASICs, we can measure the next day. 24 hours is plenty. For other proteins, we need to wait 2-3 days. And I know people that wait up to 5 days. It's up to you to determine depending on your protein. But I recommend to start checking just 1 day after injection. You might be surprised how good currents you have already.
Another question.
Sorry,
Go ahead. I'm sorry.
is that there is considerable variability from batch of oocyte to oocyte. So if one batch gives you low level of current, probably next week you will have another batch of oocytes that gives you 5 times more current, even if you inject exactly the same RNA. It's variability.
I see. For those who are interested in doing DNA, injecting a DNA, are you able to see the nucleus, or are there some specialized techniques for targeting the DNA into the nucleus?
Yeah. Well, the nucleus is under the black area of the oocyte, and it's not visible. So if you want to inject the nucleus, you can get frogs that are albinos, and they don't have the pigmentation. And the other way to make the nuclei more visible or apparent is to centrifuge at lowest speed the oocytes. That brings the nucleus very close to the plasma membrane. And then you can find it. The black pole is actually good for certain applications. For instance, for the patch voltage clamp fluorometry, is good to image the black part of the oocyte because it usually has less background.
I see. Okay, I believe this wraps up most of the questions we received. If we've missed any questions, then I apologize, and we'll follow up with you via email. Our contact information is on the screen in case you wish to speak to us. Also, you're welcome to contact Warner Instruments if you'd like any information regarding the equipment that we've shown today. And we thank everyone for attending, and we hope you have a wonderful day.